I'm Maddison Molina, a scuba diver, an avid traveler, and a lifelong ocean advocate. Recently, I discovered that right here in the US, we have one of the world's largest and most important coral reefs, the Florida Reef. The Florida Reef extends over 350 miles from the St. Lucie Inlet in the north to the Dry Tortugas in the Gulf of Mexico. Most people don't know that this amazing natural treasure exists. So, it's not surprising that even fewer know that the reef has been slowly dying for the past 30 years. In fact, over the past few years, the Florida Reef has lost as much as 98% of its coral cover. The impact of coral extinction would be huge. A rapidly dwindling supply of seafood and fish, much less breathable air, and gray and brackish oceans. Florida's coral reef is experiencing a fast-spreading and deadly coral disease outbreak that is seriously endangering the reef's long-term survival. The disease is thought to be caused by an unknown bacteria or a virus that can be transmitted through other corals through direct contact and water circulation. Disease outbreaks are common in nature and in the oceans, but this outbreak is unusual because it spreads rapidly, covers a large geographic area, has lasted much longer than usual, and is a highly effective killer of the coral species it targets. As the largest coral reef in the continental US, the loss of this reef would be an environmental disaster, devastating the thousands of species of marine life who depend on the reef for food and shelter. Also, the loss of this reef would damage Florida's economy for decades. The reef is estimated to support 70,400 jobs and directly or indirectly generates $8.5 billion in fishing and recreational tourism revenue. While visiting the Florida Keys and learning about the reef, I came across an organization that is doing amazing work to understand why corals are dying on the reef, what we can do to reverse the damage, and how we can restore the corals that have been lost. Welcome to the Mote International Research Center for Coral Reef Research and Restoration. Located in the Lower Keys, about 24 miles north of Key West, Mote Marine's Laboratories Elizabeth Moore International Center for Coral Reef Research and Restoration is a fully equipped marine science facility dedicated to marine research and education in the Florida Keys. What makes Mote unique is its science-based approach to coral restoration. While many organizations are focused on replanting corals, the scientists at Mote cultivate a variety of genetically distinct corals called genotypes to identify strains that can resist disease and other stressors. I'm excited to meet with Mote researchers and scientists to learn more about how Mote is helping save Florida's coral reef. Based on your research, what are the most common coral diseases that impact corals around the world and in Florida?
Well, historically, the Caribbean has been impacted by diseases such as white band disease, white pox disease, black band disease, and yellow band. And these diseases are named morphologically. But recently, starting in 2014, a disease called stony coral tissue loss disease cropped up starting around Miami. And since then, it's spread throughout the Florida Reef Tract and throughout the Caribbean. And that's caused massive mortality across 20 different species of coral.
Okay. Are these diseases caused by climate change effects such as warming or higher acidity in the oceans, or are there other factors as well?
They're not directly caused by environmental stressors. We know that most coral diseases we believe they have a bacterial or viral role in them because they can be transmitted from one coral to another, and also because they can be treated by antibiotics. But a lot of environmental stressors can make coral diseases worse. In particular, we know that nutrient pollution and thermal stress both exacerbate coral disease. And actually, overfishing, which you wouldn't really think would have a role on coral disease itself, can make coral disease worse because it allows for the growth of macroalgae, which covers the coral and allows the coral microbiome to shift dramatically. And that can stress out the coral and make it much more susceptible to disease.
Oh, wow. So how do you make corals more disease resistant?
Well, this is actually a project that we work on with the coral reproduction and restoration programs. So we can look at the genetic variability of corals and assess how their disease susceptibility varies. But we work with these other programs to make babies that are better at responding to disease. So if we can identify a coral that's better at responding to disease, we can select that as a parent to use in assisted sexual reproduction activities. But we can also look at the microbiomes of our corals that we have on site and make predictions about whether they might be disease susceptible or disease resistant in the field.
How do you guys determine which corals are disease resistant?
So we can actually use experimental assays in the lab to expose corals to known pathogens that cause coral disease. And that way we can see which of our genotypes or different genetic variants that we have on site might do better when exposed to disease or might get sick really easily. Why don't we go upstairs and we can check out some of the ways that we assess disease on site. So here we've got Serratia marcescens, which is a known pathogen that causes white pox disease in Acropora palmata, or elkhorn coral. It's nice and pink. Sometimes you'll actually see this on your shower curtain. If you see your shower curtain turn pink, yep, that's Serratia marcescens. It's not pathogenic to humans, but it will cause disease in corals. And actually, this strain was isolated from human sewage. So we found probatively that human sewage causes white pox disease in elkhorn coral. So what we can do with this pathogen that we can culture on site here is we can take a swab of this pathogen and grow it up in some sediment from our coral nursery, so just sand. And the sand will become coated with the bacteria. It will grow all over the sand, and then we'll sprinkle the sand on a coral fragment. And if the coral gets sick, we'll know it's disease susceptible. If the coral doesn't get sick, then we can explore more and see if it's disease resistant.
Grace's research is uncovering critical information that will help identify strains of the disease-resistant coral, which will not only help corals survive here in Florida, but will also aid in understanding how to prevent and treat coral diseases as infections occur around the world. Now that I understand how Mote studies coral diseases, I wanted to learn more about Mote's coral reproduction program. Celia Leto is a marine biologist who studies coral reproduction. Her work allows her to understand every aspect of coral reproduction. So what are the main stressors that prevent corals from reproducing?
There's a lot, and they all combine together to make this perfect storm of stressors. But the main thing that we're observing in the Florida Reef Tract right now is a combination of disease and thermal stress. Any form of disruption in their daily life or daily state can upset their ability to produce gametes, therefore they cannot reproduce. And because disease is a huge issue in the Caribbean Reef Tract right now, we're noticing that a lot of corals aren't spawning because they become sick or start showing signs of becoming susceptible to those stressors.
So how do you know in a nursery that a coral has a high probability of spawning?
So every summer, my PI, Dr. Hannah Cook, she's the head of the reproduction department, and we go out to the spawning nursery and we do something called sexual maturity assessments, or SMAs. How we do that, once again, it's species dependent, but in our branching species, we'll do something called cracking. You take a little snip on the inside of one of the branches, and you can actually see in July or August, September, depending on when that predicted spawning window is for that species, you can actually see if they're producing gamete bundles. They look like little pink dots on the inside of the skeleton, and that's how we're able to determine if they're ready to spawn and how far out we are from witnessing spawning. And then in our massive species, which are the big bouldering corals like brain corals, Montastria cavernosa, all these enormous corals. They don't have branches, but they cover a lot of surface area. So we will actually take a drill and we will core a small tissue sample, and we will look inside their skeleton and look for the same thing, whether or not we can see the presence of gametes.
After our discussion, Celia showed me how Mote studies spawning in her lab. She programs temperature, tidal patterns, and a lighting regime to reflect the exact solar and lunar cycles that the corals would experience in the ocean. They call this process assisted sexual reproduction, and it allows Mote's team to create new genetic diversity within the same species that are more resistant to disease and other stressors. By creating genetic diversity through their assisted sexual reproduction program, Mote is identifying which families of coral are best to transplant in the wild to increase their chances of rapid reef restoration. They are growing them at a much faster rate than they would grow in the ocean. How do they accomplish that? For that answer, I spoke with Summer Brooks, a research scientist at Mote, focused on operating and monitoring their land-based coral nurseries.
So we do use a different method than a lot of different places because Mote has the unique capability of spawning our own corals. So Dr. Cook and Celia here at Mote spawn our corals, and we use those sexual recruits to then microfragment in.
How long does it take to grow coral that is able to be transplanted onto the reef?
It depends on the coral species. So Acropora palmata can take 4 to 6 months to grow from a piece of Lego to a silver dollar. And then for the same size range, it can take a massive species such as rain coral 10 to 14 months.
Wow, that's cool. What things do you have to monitor each day when you're growing corals?
So water quality is very important. That's actually one of my key roles here in the land-based nursery. I monitor the temperature, pH, copper, phosphates, you name it, I monitor it. Also, pest mitigation is pretty important, and light.
How do you attach the coral to the reefs?
So we use a 2-part epoxy. We actually take underwater drills and we drill holes into the live rock. And we then use the coral plug, put 2-part epoxy on it, and stick it in the hole.
Corals that were grown in Mote's labs and transplanted onto the reef were observed spawning in the wild. Spawning is a once-a-year event for corals, where colonies release their sperm and eggs into the water column. It's a dazzling display and the first step in the sexual reproduction process.
I remember the biggest spawning event that happened at Mote was my PI, Dr. Hannah Cook. She has been observing this colony of Orbicella fabulata in 2020. It was outplanted 5 years prior, so it had survived Hurricane Irma, it had survived bleaching, it had survived disease, and we were a little worried that it wasn't going to spawn. But during its predicted window, she went out and she waited, and there was a whole camera crew there and everything, and it spawned right in front of her. So that was one of the most exciting things for her. It's really reassuring to have confirmation that the work that we're doing is going to create functional self-sustaining reefs in the future, because something that we have to consider is, if we are able to settle corals and care for them from a really young age and then get them to the point where they're adults and we can outplant them, that's great, but will they be able to function on their own once we stop?
In the wild, corals can take decades to grow large enough to be able to reproduce. But with Mote's pioneering method of coral microfragmentation and fusion, the growth process is sped up and occurred in just 5 years. So over the long run, Mote's transplanted colonies will create stronger reefs with an improved chance of surviving the stressors that are bound to come their way as climate change continues to cause ocean warming, unpredictable catastrophic weather events, and increased diseases. The Mote International Research Center is an amazing place filled with researchers dedicated to understanding and saving the delicate ecosystems of the world's coral reefs. The work they do here is amazing, and if you ever get an opportunity to enroll in one of their educational programs if you visit the Florida Keys, I highly recommend it. You'll meet some great people and learn more than you ever knew about coral and saving the Great Florida Reef. My name is Maddie Molina. Thank you for watching. I'll see you next time.